Alrighty. Whoa. It's always set up. Okay, sorry about that. The settings on this seem to be different every time I walk in. Before we get started, one of your classmates, Nathan Berg, has really taken the lead on helping put together a really amazing event tomorrow. So I'm going to let him talk about that for a second. I hope many of you show up to it on this thing.
Can you guys hear me? Yeah. Okay, so I'll just tell you a little bit about our student org. So we're Colleges Against Cancer. We're an entirely student-run org, and we partner with American Cancer Society. And if you guys know a little bit about ACS, the main fundraising event of the year is Relay for Life. So if you guys are familiar with it, essentially the main point of it is to raise money, but a big part of it is there's essentially, we keep people walking around the track during the whole event. And the point of that is cancer never rests, really. Someone who's going through cancer, they can't take a break, they can't take a day off. It's a day in, day out, struggle. And so we're not only raising money, but at the same time, we want to remember that it's an ongoing fight. And it's a good thing for us to remember that if we have a family member, a friend who's going through cancer, on a daily basis, text them, give them a call, see how they're doing. It's important that we just raise money to support really amazing research that's being done or providing rides for people who are going through chemo, but at the same time being there as emotional support. Because it's not just a physical battle, there's a lot of mental strain. So it's going to be a really great event tomorrow. We're going to have speakers. I hear there's a cancer biologist who is really good, so you guys should look forward to it. Dr. Saxena will be speaking tomorrow. There will be an oncologist, and then there will be a survivor who actually received treatment at the UI Health Cancer Center. So it's going to be a really great day. There's going to be food, games. I'd really like to see a lot of you guys there, and if any of you are interested in joining our org, we're always open for that. So if cancer is something you guys are affected by or something that's important to you, just if you see me, come talk to me after class, or if you see me in the quad anytime. But it will be at the rec tomorrow from 5 to 12. Drop in whenever you want. If you can't make it, please tell people about it. It's a great event, and I hope to see you guys there. Thanks. Oh yeah, the talks will start at 6 o'clock, so if you want to get there for the talks, they start at 6.
Yeah, so the talks are going to have, there'll be an oncologist, a cancer survivor as well, right? Yeah, and then as he said, the cancer biologist is a little bit shady. He's this guy you might have seen up here talking to you right now. But the other two, I'm sure, are honorable people who will give nice talks. So. No, it'll be a fun event. I hope you guys come to it. Okay. All right, so we're starting a new topic today. And we'll see how it goes. I've had bad luck being sick while teaching this semester, and let's just see how this goes. I mean, I'm going to try and give this lecture, but if we have to, we'll end a little bit early. We'll see. Okay, so today's topic is the cell cycle. And we can't teach you cell biology without teaching you about the cell cycle, which is fundamentally how cells go through division and their progression of different stages. It would have been kind of funny if I just kept walking, right? Just left. Wonder how long this thing. Okay. The cell cycle is shown here. This is what we'll be talking about in totality. It's the first part of today's lecture, and then we'll get to cell death if we have time, which is the yin and yang of what cells are doing. So they kind of have to. Hey guys, please. The noise is distracting, so let's try to keep the conversations down. It's almost the yin and yang of how cells behave because they have to divide. So for example, if you are a child growing, you are making trillions and trillions of new cells, which means you have to do lots and lots of cell division. At the same time, we have cell death, which is a very important part of normal cell behavior or multicellular organism composition as well, and that's driven by something called apoptosis, which we'll get to if we have time. All right, so the cell cycle. 3 major parts of the cell cycle are shown here. Part 1, cell growth and chromosome duplication. So this would be a cell, here's your nucleus. These would be strands of DNA. And the idea is that during this stage here, which we'll talk about briefly, you end up with replication, so you have twice the amount of DNA here. Chromosome segregation, where the DNA is now separated out into 2 parts of a cell, and you can start to see that the cell is probably going to divide. And then the third part of the cycle would be cell division, where you actually divide the cells, and you have 2 daughter cells that are formed. So that's a really broad overview of the cell cycle and how you divide cells. And we'll get into the details of that. These are some of the topics we're going to cover. Much of what we talk about today is going to be a review in a sense that you should have encountered this in high school biology, BIOS 100, and so on. So I'm going to give you an overview, that whatever I have time to tell you about, and then when it comes time for your entrance exams, you will probably need to go into a little bit more depth than that for those purposes. So because there's a lot of material here that we could cover that I won't cover, for this lecture in particular, I'd like to give you a very clear cheat sheet about what you have to memorize, which I'm not fond of memorization, but of course it's a required part of education. So these are some of the things that you do need to memorize for this class. And I'm not going to read through them, but here they are for you to look at later. And then these are concepts to learn. As you saw from my exam, I'm big on concepts and understanding a process as opposed to memorizing just a word. So these are some of the concepts that you want to keep in mind as we go through cell cycle stuff and then apoptosis as well. Okay, so the eukaryotic cell cycle usually has 4 phases. Before we get to those phases, if you're coming in late, please try to come in from you're fine now, but let's try to not distract me, please. Okay, so there's 4 phases that we'll get to in a second, but what I want to point out to you here is that the cell cycle time that encompasses these 4 phases varies so dramatically. So for example, if you went and looked at an early frog embryo that's dividing, you would see cells dividing every half an hour or so, over and over again. In a zebrafish embryo that we study, you can see this happen at the earliest stages in 15 minutes. So if you're really bored, you can just watch cells divide under a microscope. On the other hand, if you look at fibroblasts in culture, it might take them up to 20 hours to divide. Now what's interesting about that is that there's such a wide variety of the time of division, but if you looked at the individual processes in a microscope, they look the same, they're just slower. The stages are really similar, if not identical. The proteins that are being used are almost identical. It just takes a different amount of time. So here then are the 4 main phases of the of the cell cycle: M, G1, S, and G2. So M, sorry, I need to cough. This is going to be a real fun lecture for all of us. All right. Just Vicks again. Okay. So, 4 phases: M, G1, S, and G2. M phase can sometimes get confusing because it sounds like M is just mitosis, and mitosis is a part of M phase, but the M phase actually consists of both mitosis and cytokinesis. So mitosis is nuclear division, how you pull apart your DNA. Cytokinesis is how you actually divide the cells themselves, the overall cell, cut it in 2. Together they both make up M phase. G1 and G2 are sort of resting-like phases. We'll cover them in a little bit of detail. There a lot of preparation happens in the cell. What am I going to do next? Because this is a very active phase, and this is an active phase in a different sense. S phase is DNA replication. You're going to be dividing your DNA. So when you're dividing your DNA, or when you're going to be going through these divisions, in between you're sort of preparing for those things and doing a bunch of other stuff. Now off here to the side it says G0. That's with this crudely drawn arrow, and that's because G0 refers to cells that aren't in the cell cycle. You don't have to be in the cell cycle actively. You don't have to be trying to divide all the time. In fact, Many cells don't. And so if you're not in one of these phases, you've exited the cell cycle, then you are in G0. All right? So these are things to find a way to kind of remember the order of G1, S, G2, M. I don't really have a good mnemonic for you on that. Go science, go something, I don't know. Come up with something that helps you remember this order. That's going to be important to know. Okay, so there is a cell cycle control system that keeps all of this happening in proper order at the right time. This is an example of a few of the phases that you'll find. So here's mitosis, cytokinesis. So here you have, in mitosis, you have DNA being separated. Cytokinesis, remember the cytoplasmic division. You can see that here, these cells are dividing. And then interphase, both of these are labeled interphase, is referring to all of this. That's something I forgot to mention. G1, S, and G2 together are considered interphase. So all of this is controlled by a cell cycle control system. And that cell cycle control system asks important questions because the idea here is you don't want cells dividing and propagating their DNA and what they're up to if there's anything wrong with them. So a lot of these checkpoints are designed to catch any problems and to kill the cells off if they're not doing the right job. So for example, before you enter mitosis where you're going to be separating your DNA out, it's an important question to ask at this point in the G2 phase, at the end of it. Did I replicate all my DNA? Because if I didn't do that here, I really shouldn't be trying to separate it. Did I repair DNA damage? If I didn't, or if the cell DNA is too damaged, maybe it's best to just kill the cell. Over here, in the middle of mitosis, there are checkpoints that say things like, well, did the chromosomes properly attach to the spindle? Right? And over here, before you duplicate your DNA, you might ask, is the environment favorable? So these are just some examples. There's many other checkpoints throughout the process. But at each of these stages, as is true for just about anything we cover, the cell or the organism more broadly is double checking stuff, triple checking, making sure things are working okay. And if not, because we have something like roughly 35 trillion cells, that's trillion with a T, and those estimates vary widely, you can imagine there's a lot of room for error. In other words, if you don't like what these 10 cells are doing, let's get rid of them and make some better cells or something, or use different cells. So there's enough flexibility that these checkpoints can be used to get rid of anything that could be damaging to the organism. All right, how do you control the cell cycle? It depends on proteins called cyclically activated protein kinases, otherwise known as CDKs. So CDKs, I'll explain in a second what that stands for. And cyclin CDK complexes together trigger these different steps. So we'll cover this in the next slide. Two proteins, one is called cyclin, and the name tells you cyclin, cycle, cell cycle, right? And then CDKs are cyclin-dependent kinases. As the name would suggest, a cyclin-dependent a kinase is dependent on cyclin for its function. In other words, if this cyclin isn't here, this kinase would be off. If the cyclin binds, the kinase turns on. So cyclins then are proteins that control the progression of the cell cycle by activating these kinases. And I haven't told you yet exactly what these do. We'll get to that, but we're starting at the top. I want you to notice this graph down here. It's a rough graph of the concentration of different cyclins in a cell over different phases of the cell cycle, G1, S, G2, mitosis. Notice how this cyclin D in red goes up and stays up for most of the time and comes back down like this. On the other hand, something like cyclin E seems to only turn on at high levels right around S phase and then goes down sharply right afterwards. So these all have different expression profiles, different concentration profiles. And because you have to have a cyclin to activate a CDK, what this is telling you is that any cyclin-dependent kinase, let's say that's a cyclin E-dependent kinase, will only be active around here. A cyclin B-dependent kinase will only be active around here. So this is a control system for keeping certain kinases active at certain times of the cell cycle. When you look at this more closely, this is an example of how a couple of cyclins behave. So here we have an active SCDK. SCDK, the name, cyclin-dependent kinase, it's called an SCDK because it's dependent on S-cyclin, right? M-CDK is a CDK dependent on M-cyclin. So they're named pretty simply. In this case, you have this active complex. Notice what happens to, let's take a look at the concentration in blue of S-cyclin. So S-cyclin, you don't have much of it, and all of a sudden goes up really sharply here and stays high during these phases. So it's turned on basically near the beginning of S phase. And so the S-CDK is then turned on basically in S phase and stays on for as long as this cyclin is there. Whereas M-cyclin, shown here, turns on much later, right before M phase, and then stays on and drops off really sharply. Notice something here. The turning on of the protein levels is more gradual, at least, and particularly in the case of M-cyclin, than the drop-off. It's really steep here, right? It slowly turns on and then drops off really sharply right there. So to turn something on, you will be either making more RNA and making more protein, or just making more protein, or taking existing protein and modifying it to activate it. Those are things we've all covered in the past. But how do you quickly get rid of something when it's already there? You want to be really quick and fast and precise about that, because you need to get rid of it for the cell cycle to know that that stage of the cell cycle is over. How does that happen? It happens through something called proteolysis. So we'll talk about that. Proteolysis is mediated by an organelle that you may or may not have learned, I don't know, in the first half of class, called a proteasome. I'm sure you probably did. Did you guys learn about the proteasome already? No? Okay. So the proteasome is a protein complex shown here with all these squiggly lines and colors that contains proteases inside of it that break down proteins that have been tagged with something called ubiquitin. So if you looked in here, if you took this and you turned it 90 degrees and you looked down it, it would look almost hollow inside of here. It's like a little cone, or a cylinder, I should say. And in that cylinder are a bunch of enzymes hanging out that are called proteases. So remember, if it ends with ASE, it's an enzyme. A protease is an enzyme that breaks down proteins. So this thing is chock full of basically enzymes that chop up proteins. Sort of like like a paper shredder is the way I think about it. And if you put anything into that paper shredder, it's going to get shredded. So you don't obviously want every protein going in there. So how do you decide what gets chopped up and degraded this way? You tag proteins that are going to be degraded. So here's a protein that you want to degrade. You tag it with something called ubiquitin. Ubiquitin subunits are hanging out, and in a process that's outside the scope of this class, or this lecture today, ubiquitin can be added by various enzymes to proteins that should be degraded in response to other signals we're not getting into. Once that tag is there, then the proteasome recognizes that tag and lets them in. And once you get in there, so here's this right here is this. So here's the protein with the tag that got in there. Once it's in there, the proteases that are inside will chop it up and spit out the peptides that'll get reused. So if you use this process, then you've got a bunch of this M-cyclin, and if you start tagging the M-cyclin with ubiquitin, it will get degraded really quickly and fall off in concentration. So that's how that works. Any questions so far? I have the candy. Okay, no questions. It's pretty straightforward so far, I know. Okay. So this is just showing you what I already told you, just an example. This is an active cyclin-CDK complex. The CDK is active because the cyclin is bound to it. If you ubiquitinate, as shown here, the cyclin is degraded, and once it's degraded in the proteasome, now you have the CDK unable to function. Okay. So the activity of these complexes depends on our old friends phosphorylation and dephosphorylation. We're back to adding and removing phosphate groups, some of the same stuff you've heard about so many times. And this is an example. You may recall I told you that while usually phosphate groups being added turn things on and removing them turns things off, I did tell you there were some exceptions to that. This is one of those exceptions. This is a case where adding a phosphate group turns things off instead of on. So in this example, you have a cyclin, M-cyclin, and its CDK, M-CDK, mitotic CDK. And a kinase called WEE1 is going to phosphorylate the M-CDK. Once it's phosphorylated, even if the cyclin shows up to activate it, it's not able to do that. This is an inactive complex. So I'm adding a level of regulation to what I've previously told you. So far I had just told you this part, right? If you ignored this, I told you, oh yeah, if there's a cyclin, it'll turn on the CDK and you're good to go. Now I'm telling you there's another level of regulation, which is that even if the cyclin shows up, if this thing has an inhibitory phosphate on it, it's not going to work. So what you're seeing is another example of multiple levels of regulation by the cell, safeguards. One level of regulation was cyclin CDK complexes are dependent on the levels of cyclin. That's what we just covered. Now you're at a second level of regulation that's not the only thing. You also have to have the CDK not being inhibited by a phosphate group put on there by a different kinase. So 2 levels of regulation so far. To open this up to being activated, you have to have a phosphatase, in this case CDC25. That's going to, phosphatase CDC25, that's going to remove that phosphate group from this inactive MCDK, and that now can be activated by the binding of a cyclin. Okay? So CDK activity can be blocked by CDK inhibitor proteins, which is a bit of a ridiculous title if you think about it. Like, okay, the activity blocked by an inhibitor. Well, yeah, that's what an inhibitor is. But anyway. All right, so this is an example of that, an inhibitor coming in and blocking the complex. So now this is a third level of regulation. I told you 2 before. Cyclins and CDKs, number 1. 2 was inhibitory phosphates. 3, P proteins. P27 in this case. So the way these proteins work is that you've got your active complex here because you've got the 2 components. There's no inhibitory phosphate. Everything's good. But wait, here comes in a P27 protein that binds to this thing and turns it off. Third level of regulation. You can see how important the cell cycle is, that there's so many different ways to turn it on and off and make sure that it only moves forward if everything is in place. All right, so the cell cycle system can pause the cycle in various ways. Remember those questions I told you about at the beginning? Did I make my DNA? Did I separate it correctly? Well, if the answer to those questions is no, then the things I just told you about come in and play a role. So for example, CDC25, that phosphatase that I told you back here, is responsible for removing the inhibitory phosphate. If you inhibit that phosphatase, now you have inhibited the enzyme that would have removed the inhibition. So you keep the phosphate group on that's inhibitory, and if you keep that on the cyclin CDK complex, you won't have active CDK and you won't move forward. That's a I know it's a little bit complicated, but if you look at these and kind of replay the lecture capture and write this down as you go, it'll start to come together pretty quickly. It's just basically multiple levels of inhibition, of activation, all the stuff we've talked about. So, but the point is that you have this checkpoint based on inhibiting CDC25. Another example here, we're not talking about APC today, or at least not in detail, I think. But basically just to understand that you can inhibit activation of proteins to keep exit from mitosis. You have CDK inhibitors blocking entry to the S phase. So this is just sort of a broad overview slide of the specific examples I gave you of how cyclin and CDKs, inhibitory phosphates, and P proteins are 3 different ways to keep the cell cycle from moving forward or to have it move forward. Okay, so let's talk a little bit more detail about G1 phase next. So G1 phase, remember, is that first phase after you've had M phase. You've divided your nuclei, you divided your cytoplasm, you've got 2 new cells, you're now in G1, and you need to start doing something as a cell. One of the things that's important is that mitogens promote the production of cyclins that stimulate cell division. So I've told you all about cyclins and CDKs, but this is the first time I'm telling you really about how cyclins are told to be made. Mitogen, you might recall from an earlier lecture, is a signaling molecule that promotes the cell cycle or cell growth. That's all it means. Mito for mitosis, you can remember it that way. Mitogen is a factor that can promote that cell cycle or that cell growth. So in this pathway here, you have a mitogen shown in red binding to a mitogen receptor and activating it. Summarized by these arrows are all the things we've talked about in the past, the signaling pathways, your kinases, etc., effector proteins. And then here's what's happening inside your nucleus. Water break. Yeah, it's a good question. Do cells have other functions during M phase, or are they just dividing? They're really focused on dividing. That's their main thing that they're doing. But that doesn't necessarily mean that they've always stopped doing other functions. So for example, it depends. So like, let's say your function is to migrate from point A to point B. You usually, for the mechanics of cell division, you have to stop that migration to divide. So in that sense, you stop that function. But when you first started the process, let's say making the proteins, you can probably still be dividing. And we'll see this actually in our live imaging experiments where cells will be moving and then they'll pause and then divide and then move again. On the other hand, if your function is something else like, oh, my job is to secrete a molecule out there so that the nearby cells get it and respond to it, then you could imagine that the cell can keep doing that even when it's, yeah. Yes, they spend more energy on dividing. Absolutely. It's a really involved process. Yeah. Any other questions? Yes. Yeah, so the question is it the amount of cyclin? The amount of cyclin is changing in the cell, but but the CDK levels, are staying pretty much consistent. And yes, that's correct. The CDKs are always there because so that they're always available at a decent level, so they're ready for action, if you will. And then you have cyclins that kind of are going through the up and down cycle. Yeah. Okay. Yes. So mitogens activate. What was I talking about? Okay, right. So so this is an example of what happens in the nucleus. So you have a mitogen promoting cell proliferation, cell growth. You have a signal that's gotten into the nucleus. And now we're putting these cyclin CDKs sort of into context in this diagram, so you can see them in the nucleus, what they're doing. This protein, RB, is a retinoblastoma protein. You might recognize the name retinoblastoma. It's a cancer that it happens in the retina, and it can happen actually quite young. It's rare, but it can happen quite young in kids sometimes. We'll cover that in more detail when we get to the cancer portion of these lectures, which is coming up pretty soon here after today. But for today, food for thought almost to keep in mind for the cancer lectures is you get mutations in cancer in proteins that are important for these fundamental processes. This is an example of that. You're talking here about cell proliferation. We know cancer cells love to divide. So it kind of makes sense when you think about it that mutations that cause too much cell division or cell proliferation will put you at risk of cancer. And think about that when we go back. Keep that in mind basically for retinoblastoma. For today, here's how retinoblastoma is involved. So active retinoblastoma protein in red here sequesters a transcription regulator and stops this process of transcription. When you activate this CDK cyclin complex, that CDK, the kinase, is going to, inside of the nucleus, phosphorylate the retinoblastoma protein, which then changes its shape. So it's like this here, and then phosphorylated, it falls away. And now this regulator in green is released and is able to bind and cause cell proliferation. So retinoblastoma protein is an important component of keeping things in check at the transcription level, because you cannot move forward in the cell cycle from G1 if you don't have the right proteins made to move forward from here to here. All right? So this is an example then of how a signal from outside of the cell is controlling a cyclin CDK-mediated regulation of this checkpoint and moving through the cell cycle using a protein that's phosphorylated by the CDK. By the way, another example of phosphorylation inactivating something instead of activating it. So just FYI. Any questions about that? Yes?
Is there like a specific mechanism that is telling the mitogen or we have the amount of proteins and stuff necessary to move on so that the mitogen will serve as the ligand?
So I think, tell me if I got the question correctly. Are you asking, so the mitogen signaling, is there something for the cell to realize that it's got enough signal to have made enough proteins to move on? Yeah, it's a good question. Is there something that lets the cell know? Sort of, yes, because what's going to happen is that you have a bunch of signal coming in, and nearby cells that are sending the signal they don't know necessarily how much signal needs to go into a cell to do this. So the cell is receiving the signal and processing it in the way that it needs to get to this point. And as long as the threshold is met, there's enough of it being made, then a lot of this downstream activation can continue once that signal has come through. The way to think about it is, I guess, more broadly, when you make enough of that protein and do enough things here to move through the phases, you've done your job. And once that happens, that movement to a phase is going to trigger the degradation of a number of proteins or just ignoring them entirely so that you focus now on what happens in the next phase. Yeah, so the signal can be quite crude, right? It's like almost like on-off in a very simple way. But the signaling pathways we've learned about, you've seen all the feedback loops and the inhibitions, and getting to your goal is the important point. And once the cell does that, then it can kind of ignore anything after that. Yeah. Any other questions? Okay. Oh, and I've tried to put these here so you can always kind of glance to see where we are in the cell cycle as we talk about different parts. So right now we were just talking about the G1 going to the S phase here. All right, DNA damage can halt progression through G1, the checkpoints we talked about. If there's a problem, you don't want to divide. So let's take a look at that. So let's imagine that X-rays have damaged your DNA. There's a bunch of damage response pathways that get activated, like, whoa, there's something wrong with our DNA here. We're not able to fix it. It's really badly damaged. And then a couple of different things can happen. So we'll talk about the repair part first. There's a very important protein called P53 that you're going to hear about a lot when we talk about cancer. In fact, you may have already heard about it because it's so highly targeted in many different cancers. P53, when there's no DNA damage, so let's ignore this for a second. P53, one of those P proteins that I talked about earlier, in the absence of any damage, it's degraded in proteasomes. So it's made at high levels and it's quickly degraded. Seems wasteful, but as we've discussed, biology often is. The idea is that the cell doesn't want to spend time making it when it needs it. It wants it there immediately, urgently available as soon as it needs it. And the way it can do that is to make a ton of it and then just chop it up until it wants to stop chopping it up, and then all of a sudden it's got that ton of it sitting there already made, processed, ready to use. So once there is DNA damage, kinases will phosphorylate P53, shown here, stabilizing it. Once it's stabilized and active, that P53 is going to go into the nucleus and bind to the regulatory region of DNA of a gene called P21. ridiculous. Okay. P53 is going to bind here and make P21 protein. P21 is one of the inhibitors I talked about earlier. So it will go in and inhibit those CDK cyclin kinase complexes, CDK cyclin complexes that I was talking about, and inactivate it. And if you inactivate this G1 to S transition CDK complex, now you're again not going forward here. So the DNA damage was detected, and this process stopped the cell from dividing that damaged DNA by using this P53, P21 mediated cascade working on a cyclin CDK complex. Now this says repair or apoptosis. That's because if you have crazy amounts of damage and you can't repair your DNA, what do? You want to keep a cell just arrested in G1 phase forever? If the damage is bad enough, P53 can also instead start a different pathway for cell death. And we'll get to that a bit later. Okay, this is mitosis. In a nutshell, it consists of 5 phases. Some of this is semantics, like these are just names that people have given to parts of the cell, parts of mitosis. Prophase, prometaphase, metaphase, anaphase, telophase. Just to give you a sense of that, when I learned this, when I was your age, prometaphase was not a thing. At that point, it was just prophase, metaphase, anaphase, telophase. Somebody decided, let's make a 5th phase in here, and we're going to specifically referring to when the chromosomes are attaching to the mitotic spindle. So it's just a matter of understanding that these are the ways, this is how we've chosen to divide up mitosis into these phases. And it's written on here are all the major things that are happening at each of these phases. Again, hopefully that somewhat is a review for you from previous biology classes. In your PDFs, you'll notice several slides after this where I have some pictures and go through it. I've taken that out for time, and also because I think this is sufficient. This is what I need you to know for this class. If you'd like to look at the pretty pictures that are there, they're in your PDFs, and learn about more of the details of this, you can on your own time. And your lecture capture has been updated so that this is the only thing we do about mitosis, and then we're going to move on from there. So mitosis then is that first part of M phase, and then I have 1 or 2 slides about the second part of M phase, cytokinesis, which is shown here. Again, cytokinesis is after you've done this, after you have taken and moved your DNA and reformed your nuclear envelope to make your 2 nuclei. You have a single cell with 2 nuclei. You have to now separate the cytoplasm or the cytosol, the whole cell out. And what happens is that the mitotic spindle, which is involved in mitosis, determines the plane of cytoplasmic cleavage. So inside of this cell, you can imagine that one nucleus is somewhere inside there and the other one's over there. And there's a spindle running in between that separated everything out. Based on that, roughly 90 degrees to it, there's going to be a cytoplasmic cleavage that happens, shown here, and zoomed in on here, a furrow that's formed. How does that happen? Well, you already learned about the 2 components that make that possible when we talked about cell migration, actin and myosin working together. So if you take these separating cells undergoing cytokinesis here and took a cross-section through there, right here, and looked in there, this is what you would see. The dark band here is because of myosin. So you have, this is a diagram of what we're looking at. The green represents microtubules that are making the spindle that separated out the nuclei, that bound to your DNA, separated it out. That's what's in green. In red, this ring is what this is a ring of actin and myosin together. And remember, myosin is a motor protein that moves along actin. And so if your actin is stuck along the sides of the cell membrane here, and you have your myosin active, then you can generate a force. So in a coordinated way, that force works, and it constricts the cytoplasm and basically cuts the cell in 2. Actin, myosin-mediated contractile ring causing cleavage of 2 cells. All right, so that completes the cell cycle for today. We're not done with lecture for today, so don't start packing up. We're going to talk about apoptosis next. But this is the first slide I showed you, I think, summarizing what we've covered very briefly. I mean, there's a whole chapter in your book on cell cycle, chapter 18. We're obviously not going through all of that. And that's why I put that cheat sheet at the beginning. For this class, we have limited time. This is what we're covering. These things that I talked about today and what I specifically put up on the screen. All right, as I already mentioned, the yin and yang of cells, if you will, cell proliferation, cell death. You make them, you kill them. So let's talk about how that happens. Apoptosis is really important for regulating animal cell numbers. Oh, yes?
I know for mitosis we always talk about the duplication of DNA, but what about the organelles in the cytoplasm? How is that divided up when cytokinesis is going on?
Yeah, that's awesome you're thinking about that. So I think they talk about it a little bit in your chapter. The question was, well, we made two nuclei and they're separated out, but what about all the organelles? You know, mitochondria, Golgi, et cetera. All of a sudden you've got half sort of divided up, right? As you're going through this process, there's massive production of these organelles happening. So some of it happens before the division, a lot of it happens right afterwards in that G1 phase. That's part of why it's that resting phase, because you've got to make the stuff you just lost. And the existing organelles are divided between the two dividing cells. In some cases, that's a random division, and other times, that's a directed division. What I'll briefly mention to you, but we won't get into now, is cell division. So when stem cells divide, for example, you are often making another stem cell and then a different type of cell that comes from that stem cell, a differentiated cell. So when you're making two different cells, the division of these organelles is a wonderful way to start making your cells different. So sometimes the division is actually not random or not symmetric, because it might make sense for this cell to get more of the mitochondria and this cell to get more of something else based on what they're going to do next. So, I mean, we're not getting into all that here, but it's actually a really nice way that cells use to separate those things out and then to build back up. Yeah. All right. Any other questions? Yeah.
Insects and mice kind of do this type of action, like we have the towel and we try to dry, that's kind of motion.
Yeah, I like that analogy. The actin-myosin-based cleavage, kind of like taking a towel and squeezing it and wringing it out. Absolutely, that's a good way to think about it. I was saying the division wasn't symmetrical. His question was about the organelles. Where do they go in a cell? So I was just saying that your endoplasmic reticulum, Golgi, mitochondria, lysosomes, they're not randomly distributed in a cell to begin with, but even if they were, you're chopping the cell in half, and if you did nothing else, you'd say, well, half went here, half went there. And what I was telling him is that that's not always the case. That sometimes it makes sense for a cell to send more of something to one side, one daughter cell than the other.
What was the sense?
Pardon me?
What was the sense?
Well, the idea is that cells have specialized functions. And when we get to stem cells in the last lecture of class, we'll talk about that when you do cell division, sometimes you make another stem cell and then you make a more specialized cell that's going to do something specific. So, for example, let's make up a situation. I have a progenitor cell, a stem cell that's wants to make muscle, but it needs to also make more of itself to make more stem cells and make more muscle later. If you have a bunch of myosin inside of you, then it probably makes sense to send a bunch of that myosin to the muscle cell so it can get started more than it does to the stem cell, if you're making that. I mean, it's a simplified, silly example, but it kind of gives you an idea. Yeah. Okay, cell death, apoptosis. Sometimes cells can self-destruct. That's called apoptosis, otherwise called programmed cell death. The programmed is a very key point. This is not random cell death by accident. This is deliberate. You're told to terminate, and that's why it's happening. It occurs in developing and adult animal tissues. One of the classic examples of this, you may have read about during development is, if you look at an early embryo, mouse, human, chicken, whatever, at the earliest of stages, they'll look really similar. And as they grow, they start to look different. One of the reasons for that is because of cell death. So, for example, we have, at the earliest stages of being an embryo, a prehensile tail, but we don't keep it. And so that tail tissue in an embryo, apoptosis, it goes away. Now, apoptosis is different from necrosis, and I'll talk about that difference in a couple of slides. Here's another example of how important apoptosis is in development and morphogenesis. This is a paw from a mouse embryo, and I think these time points are maybe a day or two apart, I forget, but maybe a day apart. The green dots here are a stain that we can do easily in the lab for cells undergoing apoptosis. So every green dot is a single cell that's undergoing apoptosis. Look at the difference in these paws. So at first you've got this webbing in between, right? And then a day later you've got this clean separation of the digits because all of this tissue was told to die off to create a properly formed paw for the mouse to use. And same thing happens to us. Again, if you look at an ultrasound and you were to zoom in on a young human embryo's limb, you would see a web-like structure, and then it slowly will separate out and you make your fingers that are separate and able to work. Okay, so apoptosis is different from necrosis. At the top here is apoptosis. This is a very simple diagram of physically what happens. Once it's told to die, and we'll talk about how that happens, the cell shrinks, the chromatin condenses, and it starts to shut down and destroy proteins. It buds itself off into tiny little fragments like this, and then those are phagocytosed. They're eaten by other cells. And as a result, it's clean. There's no inflammation. It's sort of like, you just brought a broom and you swept it up, got rid of it, and you're done. Nice and clean, everybody's happy, things go on. Necrosis is very different. Necrosis is uncontrolled cell death by accident. For example, if I was to hit my finger with a hammer and cause damage, a bunch of cells would die. If I scratch my hand like this, and probably just killed off thousands of cells that fell off, right? Or a burn or a heart attack. These are all examples of surprising, unexpected, unregulated cell death. And when you have those things happen, you have necrosis, the cell swells, and because it's not a controlled process, now it's just like chaos in there. The cell starts to leak, things are leaking out, not good for your environment. You've got lots of enzymes and things in here, and you've got lots of stuff out here that's not good for the cell. And so everything just starts to go bad, and you end up having inflammation. So if you have, we all get inflammation in different ways. For example, if a mosquito bites you, you're going to see a nice red spot there. That's inflammation because of necrosis. If you, again, burn yourself, you get tons of inflammation. Necrosis is happening. And you have, if on a massive scale, you could have massive tissue death, and that can be a big problem. That's what happens when you have, for example, severe burns, and you end up with a pretty serious situation that has to be carefully managed. So apoptosis, controlled cell death. Necrosis, uncontrolled and very unhappy cell death. So how does apoptosis, yes?
In Duchenne muscular dystrophy, the person that undergoes these cells that lack dystrophin-expressing cells, are those cells undergoing necrosis, not apoptosis, so they don't express dystrophin? Like they are muscle cells.
Well, but are they dying? They don't express dystrophin, but...
Yeah, so muscle cells are weak, so does it mean they are undergoing necrosis?
Well, so if cells aren't expressing dystrophin, but they're alive, then they're not undergoing either of these processes. If they're told to die because they're not functional, then that would be apoptosis. Does that make? I mean, if the cell's there, then it hasn't undergone cell death, right? Does that make sense? So some of the cells may be told to undergo apoptosis, but to my knowledge, necrosis wouldn't be an outcome there, at least as far as I know. Yeah. Mm-hmm. Gangrene, yeah, gangrene would be like an infection, so that would involve a lot of necrosis. Any other questions? All right, so let's cover how apoptosis happens at the molecular level. What drives this process? So apoptosis is mediated by an intracellular proteolytic cascade. Now that's a couple of big words just to say inside of the cell, intracellular, you have lysis of proteins, proteolytic. So inside of the cell, proteins are being chopped up. That's all this means. And cascade, as you know, signaling cascade. So remember this pathway I showed you, the p53 mediated pathway that responded to DNA damage. This is what I showed you before, that this is how you can do the DNA repair after inhibiting the cell cycle from moving forward. p53 is a DNA damage sensor. p53, if the damage is too bad, can also help initiate the apoptotic signaling cascade, which is not this one. So to talk about that signaling cascade, I'm going to first, before I get back to p53, introduce a type of enzyme to you called a caspase. Again, ASE as means it's an enzyme. Caspases are proteases, an enzyme that chops up proteins. And these caspases are made as inactive precursors until they're turned on by an intracellular proteolytic cascade. So this is what caspases look like, really simplified, but this is an active caspase molecule. Let's start over here. As with any protein, N-terminal end, C-terminal end, you have two inactive procaspase molecules shown here. Procaspase, the PRO, pro, usually when people say like pro in this sense, procaspase, it sort of means something that comes before the thing. So a procaspase is the precursor to the caspase. All right? A procaspase is inactive and has these different colored domains here. This is called a prodomain, hence the name procaspase, if you will, kind of go together. And what's happening here is that once this is activated, which I'll tell you how it happens, then this part here is chopped off, and there's another cleavage site here that gets chopped off as well. The gray part, the prodomains, fall away. Now you've just got these green domains that are active. And this piece, once it's chopped off, is able to fold over and bind on like this. So that you have a large and small subunit, two different molecules like this, and they come together to make a single active caspase molecule. So you go from inactive procaspase to active caspase. Once you have an active caspase, it's a runaway train situation. And that's really important. Similar to what we talked about how important it is to regulate the cell cycle and cell division, if you've decided a cell needs to die, that's a big decision, and you don't want to come back from that. You don't want to make it possible for something to, like an infection, like a virus that's trying to hijack you, or a cancer that's trying to mess with you, to counteract that. And so once you get going with this, it's really hard to come back from it on purpose. You've been told to die, you're going to die type of thing. One of the ways that it happens so quickly is to take this signal and amplify it super fast. So a single molecule of the caspase, of this caspase, X is just, arbitrary, don't worry about that, is going to now activate many more molecules of a different caspase, which is going to activate many more molecules of a different caspase. Each of them is going to degrade different proteins. For example, nuclear lamin, or a cytosolic protein. So once you get this started, once you've activated caspases, they get a runaway train of activation going that's going to lead to irreversible, not reversible, cell destruction and death. Very effective pathway. So there are two pathways that mediate this process of turning caspases on: intrinsic and extrinsic. Intrinsic inside of the cell, extrinsic from outside of the cell. The intrinsic apoptotic program is regulated by the BCL2 family of intracellular proteins. This is the BCL2 family of proteins in a very simplified diagram. There are 3 classes, and this yes? The question is, is that the only thing that caspases do in the cell is to cause apoptosis? To my knowledge, yes. I'm not sure, but to my knowledge, that's the only thing they do. Unless you want to count the minor exception of also activating other caspases. But yeah. Any other questions? Okay. So the BCL2 family is a little bit confusing in how it was named. Nothing we can do about that, but learn it. So the BCL2 family refers to the whole family of proteins, includes BCL2, which is a protein in the family of BCL2, and other proteins that are not named BCL2. So just kind of be aware of that. These are all BCL2 family members, of which just one is named BCL2. It gets a little bit further confusing because they have different functions. One of the classes, there are 3 main classes. One of the classes is anti-apoptotic. So it inhibits apoptosis. It promotes the cells to stay alive. The other 2 classes are pro-apoptotic. So they actually promote apoptosis. And these are just some of the examples of the proteins that are listed. These colors here represent different domains that are in the proteins. I don't expect you to learn the BH1, 2, 3, 4 domains and what's and what, but just it's meant to illustrate that there are 3 different classes of these proteins that look different. So I mentioned P53 a few slides back, and then I didn't bring it up again, right? I told you that P53 can be involved in apoptosis, and then I kind of just stopped. I wanted to first tell you about the caspases, and I wanted to tell you about the BCL2 family. So now we're going to bring P53 back into the picture. This is where it comes in. P53 can turn on Noxa, Puma, Bax, and so on. And these are all examples of pro-apoptotic BCL2 family proteins. And P53 can inhibit BCL2 and so on, which is an anti-apoptotic protein. So P53 is doing 2 things to the same goal. It is inhibiting the inhibitor of apoptosis, right? We're back to our inhibition of inhibition, which then promotes the apoptosis. And additionally, it's going to directly promote the apoptosis by turning on the pro-apoptotic proteins as well. So P53 really is a master regulator of getting this apoptotic cascade going. All right, so P53 affects BCL2 family proteins, and I told you about caspases earlier. Where's the connection between all of this stuff and the caspases? So that's going to bring us now to the intrinsic pathway in a slide. This is actually a really amazingly beautiful thing that happens inside your cells because it's so fast and so efficient. This is your mitochondrion. You've heard about cytochrome C before as being important for your energetic processes. So cytochrome C does its normal job. It's part of the electron transport chain and all that good stuff inside of your mitochondrion. The red dots are cytochrome C. Without anything happening related to apoptosis, it's doing its job in the mitochondrion. No big deal. But once you have a signal coming in that says, hey, it's time for apoptosis to happen, and the P53 starts turning on, activating, promoting these proteins, this is what happens. Bax, just as an example, or BAC, are proteins that hang out in the membrane of the mitochondrion. Normally they don't do anything, but once P53 activates them, what that is code for, this vague term of activation, is that P53 encourages these proteins to open up pores. And now all the cytochrome C that's supposed to be in here, there's none outside of the mitochondrion, in cytoplasm, and so there's a huge gradient, right? A bunch of it in here, none out here. You open this up and it all flows right out. So now you've started the runaway cascade of apoptosis. You have released cytochrome C from your mitochondrion outside of it where it's not supposed to be. As soon as that happens, that's detected by adapter proteins that will bind to cytochrome C, and they will, once that binding happens, they'll form this funny looking structure called an apoptosome. apoptosome, the name comes from apoptosis. This apoptosome, once it's made, is able to recruit, bind, and activate these procaspases, in particular procaspase 9. 9 is involved in the intrinsic pathway. So once that's recruited, you make the apoptosome. So I guess I misspoke. This is not yet technically called an apoptosome. It's called that once you have the caspases bound. So now you've got your now you've got your procaspases that have been recruited and bound into here with your adaptor protein and your cytochrome C. And this apoptosome now activates the procaspase and turns it into an active caspase molecule. So this is showing you a single molecule, right? But then you're kind of zooming out, and I'm showing you that that single molecule is one of several as part of this structure. Now, for all of this complicated stuff, an apoptosome only has 3 components: cytochrome C, adaptor protein, and procaspases. That's it. Procaspase 9. So cytochrome C, adaptor protein, and procaspase 9 come together to make an apoptosome, which then is activated by all of this and takes you down that apoptosis pathway that I just told you about, where more caspases are activated and you have a runaway train now of chopping things up. All right? Any questions about that?
So is the bags or bag, can you say like they are receptors?
So the question is, are they receptors? They're really more channels in this case. I don't know whether or not to consider them receptors. I would just say that they're channels that are opened up. You could consider them receptors, I suppose. I'm not sure. Yeah. Any other questions about this? Yes.
I remember from the RTK activated PI3 kinase, the AKT actually activates the BCL2. So does the P53 inhibit the AKT that actually inhibit the BCL2, or is there a different pathway?
Oh man, that's great. So you're asking about the AKT-based survival pathway that would inhibit one of these proteins, the back protein that then BCL2 maybe it was.
It would actually activate
BCL2, right? And by inhibiting that, now you can let the cell survive. So the question you're asking is, how does P53 interface with this AKT-mediated pathway? For the purposes of this class, think of them as 2 separate pathways, and they kind of are. The RTK-mediated, PI3 kinase-mediated AKT activation is all about cell survival and stopping the BCL2 from doing its job. So that's sort of, if you imagine that pathway doing its thing. Then you've got the separate pathway with P53 and DNA repair and all of that. That's doing its thing. And then you've got a separate pathway for apoptosis where P53 is coming in and activating the BCL2. So to put it more simply, the P53 pathway and the survival pathway of AKT are all, in a sense, competing with each other. And the way this is set up is that the P53-mediated apoptosis pathway should win the battle because it's irreversible. Once the P53 is able to activate the back or the backs to start this apoptosis, then any survival signal is not sufficient to override it. But getting to that point is tricky because if you are inhibiting the BCL2, perhaps you can inhibit it enough so you aren't able to have the P53 do its job. It's complicated. It's a fantastic question. There are separate pathways doing sort of the opposite things, and there's definitely a tug of war there that can happen. It also depends on context. So when we get to cancer, what you'll notice is that cancer cells will take advantage of that tug of war. In other words, they'll try to inhibit the P53 pathway to stop apoptosis, and they'll try to promote the AKT pathway to promote cell survival. Yeah. But that's fantastic. I like how you're integrating these things together. That's great. Any other questions? Okay. Okay, so that was the intrinsic pathway. The extrinsic pathway brings in signals from the outside that promote apoptosis. So, What does that mean? Well, imagine, so this was all inside of the cell, based on, let's say, DNA damage or some other issue inside the cell. But imagine that a cell doesn't want to die. It thinks it's doing fine. There's something wrong with it. Maybe it's an invading cell. Maybe it's been hijacked by a virus, whatever the case may be. You have cells in your immune system, such as lymphocytes. Their job is to kill off the cells that don't want to die on their own. And the way they do that is once they're told to go target a cell, cells express something called a fast death receptor, shown in purple here. And lymphocytes will express something called a fast ligand. And it's a cell surface bound ligand for this receptor. So it's going to activate it if it binds. Inside of cells, you have a complex that's called a DISC, death-inducing structural complex, I think. Sorry, I should know that. I'm just a bit out of it. But that's what DISC means, death-inducing something complex. It might be structural. Maybe not. Anyway, the point is that this complex down here has a couple of components to it. So you have a FAD adapter protein that has in red a death domain, which is very darkly named, and a death effector domain, which is even more darkly named. So it kind of tells you what this protein is doing. It's going to make death happen in the cell. This domain here will bind, once the receptor here is activated, then this FAD adapter protein will bind to the receptor. So the red binds to the red and the blue hangs out. That's what's shown right here. There's your adapter protein. And then this part right here in blue will bind to a death effector domain found on pro-caspase 8. This is handwritten because I fixed it. This is a typo in your book. It's not caspase 8 yet because it's not activated. It's pro-caspase 8. So pro-caspase 8, not 9. Remember, 9 was intrinsic. Now we're talking about the extrinsic pathway, and 8 is involved. Pro-caspase 8 will then bind to the adapter protein, which is bound to the receptor, and you have the death-inducing complex here, the DISC that forms. So again, this might look complicated, but it's kind of the same thing you just saw with the apoptosome. There's only 3 components here. You have a caspase, or a pro-caspase rather. You have an adapter protein, and you have a signal, in this case from a receptor, right? Basically something that's causing this to happen. So if you go back to the apoptosome, here we had a signal that was cytochrome C, an adapter protein, and then a pro-caspase. And here you have loosely defined, quote unquote, a signal, an adapter protein, and a caspase. So broadly speaking, 3 major things coming together to induce the cell death cascade to move forward. Once you have this complex, the DISC, now your pro-caspase is now activated, turned into activated caspase 8, released, and goes and does all the stuff I told you about before and kills the cell. So intrinsic and extrinsic pathways of apoptosis. Any questions about that? Yeah. Do we have other death receptors? We do. And what are the types of ligands that bind to them? I'm not sure. I think there's several different ones. In fact, there's an arms race that happens because we have lots of death receptors, and when we get infected or otherwise something doesn't want the cell to die, it'll try to mask those, and then there are other ones that can be expressed. It's a bit of an arms race. Can a ligand bind by accident? That's usually no. That's pretty carefully regulated because you don't want to accidentally kill cells off. Although you're kind of touching on an interesting topic, which is autoimmune responses, for example. Is that maybe what you were thinking of? Or yeah, right. So autoimmune responses are often a result of or by definition, a result of our immune system attacking our own cells when it's not supposed to. And that can happen because your immune system has been incorrectly trained to think that a protein that's being expressed by a cell that's actually your own, it thinks it's foreign, so it attacks. But that's more on the recognition side. In other words, it has to recognize that incorrectly that cell should be attacked. And then the cell that comes in and does the attack, I think, is more responding pretty simply to the fact that it was supposed to do that now. But I'm not sure beyond that. To put it more biochemically, the ligand binding to the receptor is very, very specific. But all the other events that control that happening are more, can be leaky. Yeah.
So does this pathway kill the same cell, or does it sense it goes to build another cell?
So does this pathway kill the same cell as or a different cell than the intrinsic pathway? All of our cells have both of these pathways possible. A cell that activates the intrinsic pathway is doing it in response to something inside of the cell. So this wouldn't really come into the picture. Vice versa, if the intrinsic pathway is not activated, then this one could be happening in that same cell because it didn't die the other way. It just depends on what's the stimulus for saying the cell needs to die, basically. Yeah. Okay, so it's pretty much time to wrap up. Don't pack up yet. I'll get you out on time. It just occurred to me that one thing I can do that would be kind of fun, since I didn't take a break earlier, is to show you apoptosis in action really quickly in a movie. And it's like a 10-second movie, so don't worry. All right, so I don't bring up too much of the work in my lab, but one of the things we do is we look at cancer cells in vivo in zebrafish embryos. And this is one of my favorite movies that I'm going to play for you. Long story short, we were able to find a way, at least in our embryos, to take human cancer cells and get them to die, differentiate into neurons, become neurons, and then die. And in this movie I'll play for you, you're going to see a cancer cell in magenta here. Inside of an embryo, it's going to become a neuron, and then poof, it dies. That's apoptosis right there. Remember I told you about blebbing? So you're seeing these little particles form. It's not uncontrolled. You actually have apoptosis causing the cell to ball up into tiny pieces that then will eventually get eaten up by other cells. So that's it for today, and I'm happy to answer any questions. Yeah? They often are, yeah. Yeah.